Dear colleagues, in the morning, I tried to share with you strategic issues unlocking the secrecy of personalized and precision medicine and PPM guided design driven bioengineering translational applications. Here is now. It's just an example what have been done by the international group. In this context, two global projects are being performed. One regarding usage stem cell. The second, it's antibodies with proteolytic activity. Unfortunately, 2 days ago, the first project is not as approved by FDA. This has been approved, and in 3, 4, 5 months, 3 companies will take it into the manufacturing. Might be in June the next year. I will present another project. Development of autoimmune inflammatory diseases is just the illustration of the concept. Is a consequence of lack of self-tolerance, which can arise due to defective function of any of the features contributing to the unresponsive status of the immune system towards self or outer antigens. So, autoimmune or autoaggressive doesn't matter. Responses resemble normal immune responses to pathogens as specifically activated by self-antigens or autoantigens and give rise to autoreactive effector cells and to autoantibodies against the self-antigens. When the reactions to self-tissues do occur and are then improperly regulated, they cause a variety of chronic syndromes called autoimmune diseases. Meanwhile, myocarditis can be defined as the inflammatory process affecting the muscular tissues of the heart, myocardial. Balancing between autoimmune myocarditis and dilated cardiomyopathy. And then it's being transformed in a stepwise manner into dilated cardiomyopathy. Okay, myocarditis is considered an important cause of sudden death, as has been shown by autopsy-based studies in the general population. Regardless of its etiology, the acute inflammation may progress to subacute and chronic stages, and finally to tissue remodeling, fibrosis, and loss of myocardial architecture and contractile function. The latter in a process of chronic damage corresponds to development of dilated cardiomyopathy, DCM. The gold standard for final confirmation of causative reason of AIM is the endomyocardial biopsy, very invasive procedure. At this point, new targeted therapies for autoimmune and inflammatory diseases would require greater understanding of a patient or a person at risk to get the therapy individualized for either of those subsets, more precisely for specific biomarkers as the target, as the potential targets. In this sense, the identification and implementation of diagnostic, predictive, and prognostic biomarkers is just the major types of biomarkers: predictive, monitoring, diagnostic, susceptibility, and related. Remains the holy grail of platforms, algorithms, and protocol, which are the crucial for PPM and PPM guided clinical cardiology. Biomarker enable pre-early diagnosis, guide targeted therapy, and monitor the activity and therapeutic responses across the diseases. We would need to discover newer and newer biomarkers and to create simultaneously, fundamentally, not clinically, new strategies based upon subclinical recognition of those biomarkers long before the disease clinically manifests itself. Among the best validated predictive biomarkers are autoimmunity-related ones. Meanwhile, the utility of predictive, diagnostic, and prognostic biomarkers to monitor and to manage chronic autoimmune inflammation and to predict stepwise transitions is dependent on three key parameters, which must be carefully assessed and only after FDA approved: sensitivity, specificity, positive predictive, and prognosticative values. That's the motivation why biomarkers fail today to reach the clinic. It sounds strange, but last year, due to the assessment of FDA, 97.7% of all biomarkers proposed failed to get validated. So, accurate prediction is crucially vital for prevention of autoaggression, and the targeted treatment could thus be given to those individuals who are most likely to develop the disease. AIM. Very brief but constructive features. A grand role in the development of autoimmunity in AIM, in particular, is the exposure of self-antigens which are encrypted and unavailable to the immune system under physiological, healthy conditions. Cardiac myosin, CM, is one of the most important self-targets in AIM. Cardiac myosin, CM. The latest concept and achievements demonstrate that the anti-CM autoantibodies play a significant, moreover, crucial role in the development of cardiac autoimmunity. The primary damage in AIM progression and evolution, which is mediated by anti-CM autoantibodies to trigger a release of separate and pathogenically valuable cancer cardiac associated epitopes into the bloodstream. Those epitopes acting as sensitizing factors to generate autoantibodies more and more, which in turn would drive the autoaggression and thus the disease regression. At present, a spectrum of cardiac-associated autoantibodies occurring in AM patients and pre-illness persons at risk has been confirmed to be very large. Including different families of circulating lipids. Several groups have shown that a subset of patients with AIM and their symptom-free relatives has circulating heart-related autoantibodies. Those antibodies are directed against multiple antigens, some of which are strictly expressed in the myocardium. For example, organ-specific for the heart. Others are expressed in heart and skeletal muscle-specific. Antibody of IgG type, which are shown to be cardiac and disease-specific for AIM, can be used as reliable markers of autoimmune pathogenesis for identifying patients, mostly only patients, not pre-illness persons at risk. For identifying patients in whom immunosuppression and immunomodulation therapy may be beneficial and their relatives at risk, relatives only. Moreover, some of those autoantibodies may also have a functional role in patients, as suggested by in vitro data, as well as preliminary clinical observations. Though further work is in progress to clarify this important issue. Along with canonical antibodies, which are widely broadly known to the scientific and clinical community, some of the families proven to occur are antibody possessing armed with catalytic proteolytic activity, and thus belong to antibodies with a feature of functionality. The examples of antibody proteases are IgG, found in a series of autoimmune disorders, to cleave targeted autoantigens and thus to act as informative biomarkers to monitor both clinic and subclinical causes of the disorder. The unique clinical case is a family of antibody proteases detectable in AIM to cleave cardiac myosin. The activity of antibody proteases, I mean cardiomyosin targeted, markedly differs between AIM patients and healthy control. Of great interest is the evolution of antibody-associated proteolytic activity at different stages of the disease, I mean AIM progression. For those purposes, we have been monitoring classical AM patients with full-scale clinical manifestations, their direct relatives and healthy controls, voluntaries, for around 2-3 years to have them tested for CM targeted antibody proteases. Look, AM patients, anti-CM antibodies, activity of proteases in AM patients with full-scale clinical manifestation. Relatives of AM patients, you see the difference? And healthy controls, you see the difference? The overall number of the person tested, about 6,000 for 3 years. Two-thirds of AM relatives, not patients yet, being initially seropositive for anti-CM autoantibodies and low-active antibody proteases, and monitored, have been demonstrating stable growth of the antibody-associated proteolytic activity during the time after the study. Look, the results is absolutely evident. Subclinical, no clinical manifestations. Clinical, full-scale patients. So, roughly, pre-illness persons at risk, patients. We just requested our partners at London's business school if we provide preclinical monitoring and clinical monitoring, each relative with subclinical manifestations and further clinic patient, each would save from $22,000 to $34,000. It's just one. I don't keep in mind now how much patients in the world now, but it's just one. Gulf area, Iran, US, Russia, and not China, Hong Kong. No, it's China, but I don't consider China, it's Hong Kong for me. Look, the activity of antibody proteases is first registered at the very early, pre-early subclinical stages of AM. Regular, non-functional antibodies are not registered at subclinical stages. When antibody proteases are low active, the inflammation is still minimized and the manifestations are thus moderate or absolutely absent. Initially, in most of the relatives, direct relatives. I guess you understand the difference between relatives and direct relatives, brothers, sisters. In half a year, you see, in a year, in 2 years, in 3 years. That's healthy control. No dynamics. As it's broadly known, canonical autoantibodies play neither predictive nor discriminative role to affect the subclinical of AIM. In contrast, antibody proteases being bifunctional. What I mean, bifunctional. The first function is binding antigen with antibody. The second, catalytic activity. Have proved to be greatly informative and thus valuable biomarkers to monitor AIM at both subclinical and clinical stages. Therefore, the proposed predictive value of CM-targeted antibody protease for the development of AIM is being challenged. The translational potential of this knowledge is in the rational design of new diagnostic tools and new targeted therapeutics based on principles of artificial, not natural, of artificial biocatalysis and biodesign. Now, approximately 2 years ago, a new amendment has been made on artificial biocatalysis, biodesign, and biointerface cooperation principle. In this sense, targeted antibody proteases can be programmed and even reprogrammed to suit the requests and standards of regeneration. Targeted antibody-mediated proteolysis today and might be tomorrow could be applied to isolate from Ig molecules catalytic domains from FAB fragment directed against cardiac autoepitopes or domains containing segments exerting targeted proteolytic activity. Antibody-based therapeutics have entered the center stage of drug discovery as a result of a major shift in focus of many biopharma companies. Efforts continue to build better therapeutics based on monoclonal antibodies. Regarding catalytic antibodies, monoclonal antibodies have been constructed in Japan, in US, and in Heidelberg, Germany. So antibody protease engineering, not antibody engineering, antibody protease engineering would offer the ability to enhance or alter not only sequence specificity, but natural functionality to be modified to expand the clinical utility of the absolutely new tools. Of tremendous value in this sense are antibody proteases directly affecting the physiological remodeling of tissues with multi-level architectonics. For example, cardiac muscle and myelin, while securing the requests and standards of regeneration. So, further studies of antibody CM degradation and other targeted antibody-mediated proteolysis may provide biomarkers of principally new generation, in addition to biomarkers, potential target candidates, and new families, and thus supplementary tools for assessing the disease progression and predicting disability of the patients and pre-illness person at risk. Thank you so much for your patience.